to next speaker, Bettina Schwab from UKE Hamburg, and she is also working with tACS, I think mostly using EEG, but also doing a lot of computational modeling work, and looking forward to your presentation.
So I want to follow up on the topic of tACS and connectivity modulation. So let me first introduce you the experimental study that we published already, and which is the basis for the modeling that we do. So here you can see how we targeted 24 subjects that were healthy here with dual site tACS and these two montages. So the trick here is that stimulation conditions only differ in the relative phase of the two applied currents. In phase means that the current is completely the same for the left and the right, whereas anti-phase means that the magnitude of the current is the same, but there is a phase offset of pi. Then we also had a third condition where the phase offset was just jittered all the time. We also did some very basic E-field simulations that you can see here to show that the field is rather focal and that the magnitude of the field is comparable for in-phase and anti-phase simulation. So the idea is that for in-phase, the magnitude and the direction are the same for the two hemispheres, whereas for anti-phase simulation, only the magnitude would be the same, but one hemisphere sees a phase reverse field. And by that, we try to modulate connectivity. And I think it has already become clear in the last talk that it's not possible to analyze online effects of tACS during EEG, but here we only look at the after effects and compare them to the baseline. And in particular, we are interested in the condition differences. This is here, it has the advantage of modulating specifically connectivity and not the oscillations in general by tACS. And it has the further advantage of having comparable side effects between these conditions. If you compare tACS to sham, for example, you will always see phosphenes, or in many cases see phosphenes, and also some tactile sensations that are not comparable, also when you compare tACS at different frequencies. So I think that's quite nice here, and we can look at the difference, for example, between in-phase and anti-phase stimulation. First, we look at the EEG data on sensor level, and we compute the average alpha imaginary coherence between all the pairs of electrodes, and then average this over all pairs. And we can see that there is a significant difference between in and anti-phase stimulation, which is not that strong, but it's there, it's significant, and decays over time. We can look at that in frequency space and find that the effect is maximal at the stimulated frequency, so 10 Hertz. And when we project the data to source space, we also find that there is some sort of space-specific effect. So the connectivity from each region to each region, as depicted here in the adjacency matrix, and the dashed box are those connections between the stimulated regions. So between the right stimulated regions and the left stimulated regions. And we can see that many of these connections did become significant, although not all of them. So our aim for the modeling is to find a possible mechanism for these results. And spike timing dependent plasticity has been suggested to be a general mechanism of after effects of tACS. So that could be a possible candidate that we might investigate here. Then second, if we do have a candidate mechanism, we might want to make predictions and further experimental results. For example, with different tACS frequencies or different application sites that might have a different delay. So for that, we used a quite small and simple network model. It has 1,000 neurons in two populations, so in total 2,000 neurons that were of the Zikevich type. So kind of integrate and fire with an adaptation model, which makes them almost as biologically realistic as Hodgkin-Huxley models. The neurons in these two populations were not coupled to other neurons of the same population, but they coupled to 100 random neurons of the other population. So there is connectivity between the populations, which is what we want to modulate here. And the connections are excitatory and have a fixed delay D. Here in this model, the delay in both directions was always the same for simplicity. All the neurons received a tACS input, which is just a sinusoidal current, but there can be a phase lag between neurons of population 1 and neurons of population 2. It's important that the firing rate distributions of these neurons is kind of realistic. So we adapted the parameters of each neuron to make it spike at a certain frequency, where the median was about 8 Hertz, and roughly that's also the average, maybe a bit lower, so 7 Hertz. When we average all the transient brain voltages and then compute the spectrum, this is kind of comparable to the LFP. And we see here that in this model, it's rather flat, so there's no prominent oscillation. So here the idea was that the network might just entrain to any tACS frequency that you apply within the physiological range. It doesn't mean that this is realistic for cortical dynamics, but it allows to test a lot of different tACS frequencies. And now critically, we introduce STDP to these synapses between the two populations. Spike timing dependent plasticity means that if you have a presynaptic and a postsynaptic spike, then the weight of the synapse will be updated dependent on these two timings, and in particular, the difference between the timings. And this is a Hebbian rule, which means that if you first have a postsynaptic spike, first a presynaptic spike and thereafter a postsynaptic spike, this is kind of a causal interaction. So there's a potentiation of the synaptic weight. Whereas if you first have a postsynaptic and then a presynaptic spike, you get a depression of the weight. This general rule has been found in different experimental studies, but we took the concrete parameters from one study. But we know that the important facts here, in particular, that A+ is larger than A- and tau+ is smaller than tau-, have been reproduced in a lot of different settings. So, for example, we can already make predictions what will happen to this synapse when we have completely random firing. Then spikes could happen at each time leg here. And if we integrate over this whole rule, we get a negative value, which means that if we just have random firing, then the synaptic weight should decrease. In contrast, if you have synchrony around 0, so you're within a range, which is smaller than the decay here, so smaller than tau+ or tau-, we will on average get a potentiation because A+ is larger than A-. So probably synchrony around time leg 0 will lead to a potentiation, whereas other types like irregular spiking would lead to a depression of synaptic weights. We can make this a bit more concrete by looking at some examples. For example, with a very short delay of the synapses. Without tACS, we said that our model would reproduce random firing in both populations, and the synaptic weights, as we discussed in the slide before, would just decrease over time. If we now apply tACS in phase, both populations would lock to this tACS cycle, and because we have the synchrony at around the same time, we get a net potentiation of the synapses. In contrast, anti-phase stimulation would lead to a depression of the synaptic weights over time. You might say that this entrainment that we see here is maybe a bit too much, or only a few neurons might lock to the tACS cycle as much as these do. So we could also lower the tACS current to make this a bit more realistic, but here we only want to show the principle. So we're assuming that neurons in general do lock to tACS, and this might now be specific to the subpopulation which experimentally would lock to tACS. So then having more neurons that would not lock would only lead to more noise in the system. So we can look at this a bit more systematically. For example, again, for 10 Hertz tACS, but with a bit longer delay. We now simulate not only in phase and anti-phase, which would be phase lag 0 and pi, but also at different phase lags. And we find that the connections from neuron 1 to 2 are most potentiated if we have exactly the phase difference, which is the time lag between these of these synapses, so 10 milliseconds, and of course the other way around too. And there are two cases where we have a symmetric model, which is in-phase simulation and anti-phase simulation. And because typically in experimental settings, we're not only interested in one direction, so we want to have a bidirectional connectivity change, which means that we typically want to simulate either in phase or anti-phase. And that's also what we did in our experimental study. So from now on, we just look at these two cases. And in particular, we will look at the difference between in-phase stimulation and anti-phase stimulation. We can do this for different tACS frequencies and different synaptic delays. And we find that for low frequencies and short delays, we get a nice effect. So nice here means that in-phase stimulation would push synaptic weights or increase them compared to anti-phase stimulation. But when you increase these parameters, you can see that the effect reverses. So anti-phase stimulation would actually lead to more connectivity compared to in-phase stimulation. And then when we have a synaptic delay, which is around 1 cycle length, of course, the effect would reverse again. So these are quite concrete predictions on like different tACS frequencies and synaptic delays. But we also want to validate the model if it's actually fitting to our experimental data. So let's look at the 10 Hertz case again. For the 10 Hertz simulation, we find that this effect, which is in-phase simulation minus anti-phase simulation, decays with a synaptic delay within the physiological range. So we said that roughly 30 milliseconds is kind of the maximum that we might have for cortical-cortical interactions. And now to compare this to our experimental data, we looked at the difference in functional connectivity, again, in minus anti-phase stimulation, and correlated it with a track length. So the fibers from each region to each region. This has the idea that the track length should somehow relate to the synaptic delay. And of course, it would be better to have the synaptic delay between all these regions, but it's really hard to get this for human data. So yeah, if we had that data, we would use it, of course, but as a first proxy, we would take the fiber length. And we do find this negative relation, meaning that the shorter the path between two regions, the nicer our effect. And we also see that most of these effects here are in the positive range. So also for a lot of physiological delays around 8 or 10 milliseconds, we do find a positive effect here. So in sum, this is okay with our model. Now we can check how we can manipulate the model to still be in line with the experimental data. For example, let's say we simplify the rule to be symmetric. So A plus is equal to A minus and tau plus is equal to tau minus. This is not realistic. It's not what experimental studies showed, but it would make the computation a bit easier. And we do find that there's still this negative correlation here, but none of these values is actually positive. So in each case here, anti-phase simulation would lead to more connectivity compared to in-phase simulation. So it's not in line with our experimental data. Then furthermore, we can look at some different network architectures that might be a bit more realistic. So we had subpopulations within each population where 800 neurons were excitatory and 200 were inhibitory. And the intrinsic connectivity here with delays leads to a spontaneous alpha oscillation. So also for the other population, which has a similar alpha oscillation, and then these are weakly coupled. So again, we have the coupling between the two populations, but this coupling is not dominating the dynamics. So the alpha dynamics is actually due to the intrinsic coupling within each population. And now the spontaneous alpha oscillation would just log to the externally applied 10 Hertz tACS. And when looking at the difference between in and anti-phase stimulation again, we find something which is really comparable to our experimental data. We can go a bit further and, for example, include connections from excitatory to inhibitory subpopulations, and we find pretty much the same. So to sum this up, SDP would be a possible mechanism of our experimentally found connectivity changes, although we cannot show that's the only one, but it would be in principle in line with the findings if we choose a realistic SDP rule. But if we, for example, change the frequency, in particular to a higher frequency, then it's not sure that we get the same result. For example, applying 40 Hertz or 50 Hertz tACS in an antiphase might not lead to the same outlasting changes. So this doesn't mean that for online changes, we might get a problem, because online entrainment could be dominant and not the plastic changes between of synapses. So we suggest before you do an experimental study, which is based on after effects and connectivity modulation, that you kind of roughly try to estimate if this should be successful and in which direction you expect the effects by having such a simple model. So I want to thank everyone who's involved in this study, and thanks a lot for your attention.
Thank you, Bettina. It was a great talk. Few questions that we have so far. First, you demonstrate that there are dependency of effects from the synaptic delay, right? What is the realistic value or range of value of synaptic delay you can expect in human neocortex? So what values you would see there?
Yeah, so it highly depends on the system. So the realistic range might be from 1 to 30 milliseconds, roughly. That's what we use here. But for example, for the motor system, say from M1 to M1 of each hemisphere, you might get a delay of 8 milliseconds. So this would be here. So say we have our experimental data, not of the motor system, but between hemispheres here, yeah, the delay could be around 6 to 10 milliseconds.